Hello, everyone, and welcome to today's webinar in the CERDP and ESTP webinar series. My name is Rula Deeb. I'm a senior principal at Geosyntac Consultants in Oakland, California, and the coordinator of the series on behalf of CERDP and ESTCP. The webinar will consist of a brief overview of CERDP and ESTCP, followed by the technical portion, which features Department of Defense funded research on using advanced molecular biological tools to assess a biological treatment of groundwater contaminated with chlorinated solvents. First, Dr. Allison Couples from Michigan State University will describe her research efforts to develop innovative methods to quantify microorganisms which degrade chlorinated solvents and 1,4-dioxane. Allison's presentation will be followed by a Q&A session. Second, Dr. Mandy Michelson from the U.S. Army Engineer Research Development Center and Dr. Kate Kusharzik from Battelle will discuss the use of biomarkers to more accurately estimate in situ biodegradation rates of chlorinated solvents. We will conclude the webinar with a longer Q&A session. On this slide, we have provided a few suggestions in case you experience difficulties with the broadcast platform. Firefox, IE, or Edge are the most compatible browsers to use with Zoom. If you lose the content on your screen, or if your screen freezes, try keying Control F5 to perform a hard refresh. If you are accessing the audio through your computer, click the arrow next to the Join Audio button, select Test Speaker and Microphone, and follow the prompts as they appear on your screen. If you continue to experience difficulties, please call into the conference line shown here. You may also submit a comment using the chat box. Please use the chat box only for comments related to technical difficulties. The Q&A option should be reserved for questions for the speakers. Today's broadcast will be listen only. You may submit questions by using the Q&A box on your screen. You do not need to wait until the Q&A period to ask your questions. We do encourage you to submit questions well in advance of the Q&A sessions. When you do submit your questions, please make sure to add your organization name at the end of your question so that we can identify you. With that, I would like to introduce Dr. Andrea Leeson, who is the Deputy Director of CERDP and ESTCP, as well as the Program Manager for the Environmental Restoration Program. Andrea has been with CERDP and ESTCP since 2001, and before that, she served as a research scientist at Battelle Memorial Institute, where she conducted research on in situ bioremediation and the design and implementation of innovative biological, chemical, and physical treatment technologies for site remediation and industrial wastewater treatment. Andrea received her doctorate in environmental engineering from the Johns Hopkins University. Andrea?
Thank you, Rula. Slide 9. What I'd like to do today is give you a little overview about CERDP and ESTCP for those of you who don't know the programs. And we have our two sister programs. CERDP is the Strategic Environmental Research and Development Program, and this is really our science and technology. We conduct it in partnership with the DOE, EPA, and DOD, and the knowledge and technologies that are developed under CERDP generally, once they're successful, can mature and go into our ESTCP program, the Environmental Security Technology Certification Program. And this is where we do demonstration and validation of mature technologies in the field. Slide 10. We have two main drivers for the type of research we fund. The first is we are looking to sustain ranges, facilities, and operations on DoD bases. This can take a lot of different formats, looking at from maritime sustainability to noise issues, sustainable forward operating bases, as well as munitions constituents. It's a very broad approach. Slide 11. And we're also looking at the bigger issue of reducing the current and future liability. We look at contamination from past practices, and that's a lot of the work that's in my program area of environmental restoration, which can include contaminated groundwater, soil, sediments, as well as a large UXO liability. And we're also looking at pollution prevention or the elimination of different hazardous materials in the whole process in the DoD for manufacturing, maintenance, and operations. Slide 12. So it's very important to us that the research and technologies we develop get out to the end user and are useful. If we don't transition this information to end users, then really the research has not been fully valuable. Slide 13. We do this in a variety of ways. We have videos that we've developed, in-person training. We have developed an EnviroWiki, as well as various webinars, guidance manuals, etc. And the webinar series that you're participating in today is one of the means for transitioning the different knowledge and technologies that have been developed under CERT of NESTCP. Slide 14. We do this webinar series generally about twice a month, and as you can see from the slide, we have a number of different webinars coming up. Registration is open for all of these webinars, and I encourage you to go to our website to get more information about these different webinars. Slide 15. You can register for the webinars at this link. You can also get archived webinars going back several years now, and you can get the audio and the slides for those older webinars too. Slide 16. I'd also like to mention our CERT of NESTCP Symposium. If you haven't been to this, it is an excellent way to see the different types of research we are funding. Everyone we fund presents a poster at this symposium, and then we also have some platform presentations with a focus on various topics. It will be held the week after Thanksgiving, December 3rd through 5th at the Washington Marriott Boardman Park, right in DC. Registration is now open, and if you go to our website, you'll be able to get information about the symposium and register for the conference. So at this point, Rula, I'll turn it back to you.
Thank you, Andrea. And it is now my pleasure to introduce our first technical speaker, Dr. Allison Couples, who is an associate professor in the Department of Civil and Environmental Engineering at Michigan State University in East Lansing. Allison's current area of research involves the use of molecular methods to quantify microorganisms involved in contaminant biodegradation. She has served as the PI on several grants to examine microbial communities associated with the biodegradation of contaminants like 1,4-dioxane, RDX, chlorinated solvents, and pharmaceuticals. And the methods she has relied on have included quantitative PCR, staple isotope probing, and high throughput sequencing. And you'll hear about some of these tools today. Allison earned a bachelor's degree in environmental sciences from the University of East Anglia in England, a master's degree in natural resources and environmental sciences from the University of Illinois, and a doctoral degree in environmental engineering and science from Stanford University. Allison, please proceed.
Thanks, Rula and Andrea, for the opportunity to give this webinar and for funding this research. So this is the agenda. I will start with the overall objective. Then I will move on to describe the various steps for the development and the validation of our LAMP assays, with the acronym LAMP standing for Loop-Mediated Isothermal Amplification. The steps involve primer design, as well as exploring the impact of humic acid on amplification. Following this, I will talk about our experiments comparing LAMP to quantitative PCR using DNA extracted from groundwater from multiple chlorinated solvent sites. I will also talk about our experiments with a visual-based LAMP assay, and the last part of my talk will focus on the use of shotgun sequencing to examine the abundance of key biomarkers involved in contaminant biodegradation. Finally, I will wrap up with a list of key points and conclusions. So the overall technical objective was to develop novel methods to detect key biomarkers associated with the biodegradation of a set of groundwater contaminants. And as I mentioned, this involves developing and validating LAMP assays, exploring biomarker quantification without DNA extraction, and creating a potentially field deployable quantification approach. And again, the last part of my talk will describe our work examining the applicability of shotgun sequencing to quantify key biomarkers in groundwater. So just a brief background slide. Lewis-McChord is of course a vital microorganism for the conversion of the chlorinated solvents to ethane. Its occurrence in contaminated groundwater is frequently investigated using molecular biological tools. These tools commonly quantify VCRA and BVCA, genes encoding for vinyl chloride reductive dehalogenases. TCEA, the gene encoding for TCE reductive dehalogenase, is also frequently targeted. As part of our research, we quantified these biomarkers using the GeneZ analyzer, as shown on the bottom part of the slide. This is a device developed by a co-PI on this project, Syed Hashim, also in the Department of Civil and Environmental Engineering here at Michigan State. So some advantages of LAMP, why we selected this method for the work. It has a high amplification efficiency, with the product being generated quickly, so this can reduce reaction times. Probably the most important advantage is that amplification occurs at one temperature, unlike quantitative PCR, which cycles through different temperatures. And because of this, an expensive thermal cycler is not needed. Also, analysis without DNA extraction is possible. LAMP can be less sensitive to substances that inhibit qPCR. And lastly, the assay involves 6 primers and therefore has a high level of specificity. The first step in this work was to design a series of LAMP primers for each target gene. For this, we targeted the reductive dehalogenase genes, BVCA, VCRA, and TCEA. We then tested the primers with DNA extracted from mixed cultures. For each primer set, we selected those that provided the most consistent results at low concentrations of our target genes. We used cultures commonly used in bioaugmentation, SDC9 from Aptim and KB1 from Serum, both containing Lewis-McChord. For the primer design, we used Primer Explorer. This is a commonly used LAMP primer design software. The image at the bottom simply illustrates an example of the 6 LAMP primers for VCRA. This slide shows a comparison of the newly designed LAMP assays to quantitative PCR. Both assays were performed on a Biorad thermal cycler and used DNA extracted from the culture KB1. The x-axis is the gene concentration as determined by LAMP, and the y-axis being the gene concentration as determined with quantitative PCR. The graph on the left is for VCRA, the one in the middle for BVCA, and the one on the right for TCEA. And the dashed line in each case represents a one-to-one correlation between the 2 methods. You can see that overall, both methods generated comparable results. We also compared quantitative PCR to LAMP using 2 different detection methods. This time we used DNA extracted from groundwater at a site previously bioaugmented with SDC9. We compared qPCR in the Biorad to LAMP in the GeneZ. And as I mentioned before, the GeneZ is a device developed to be a low-cost analytical instrument for LAMP. LAMP in the GeneZ is shown on the x-axis, and qPCR in the thermal cycler in the Biorad is shown on the y-axis. The results for VCRA are in blue, and those for TCEA are in red. You can see, in general, the 2 methods produce similar results, with the LAMP GeneZ generating higher values for some samples. The results from our experiments examining the impact of humic acid concentration on qPCR and LAMP are shown on this slide. Here, each reaction was spiked with a fixed amount of RDHA obtained from the mixed culture TCA20 from Aptim. On the graph, humic acid concentration is on the x-axis, ranging from 0 to 300 milligram per liter. The left y-axis indicates the time required to achieve a signal above background or threshold time for LAMP. The right y-axis indicates the number of qPCR cycles to reach a signal above background. The black circles represent the results from LAMP, and the red triangles represent those from qPCR. You can see at the lower humic acid concentrations, both methods provide consistent results. However, at higher humic acid concentrations, qPCR was inhibited, whereas LAMP was not inhibited. Eventually, at 100 milligram per liter, LAMP is also inhibited. An additional set of experiments focused on examining the impact of humic acid on different concentrations of the target gene when using LAMP. For this, we used a high concentration of humic acid and a range of RDHA gene concentrations. On the graph, the gene concentration is on the x-axis and the LAMP threshold time on the y-axis. The green symbols did not receive a spike of humic acid, whereas the red symbols received 30 milligram per liter. Over the majority of gene concentrations, humic acid had little impact on the threshold time. However, inhibition was noticed at the lowest RDHA gene concentration, as circled on the graph on the left. These results indicate that care should be taken when quantifying with high humic acid concentrations and low gene copies. Following the successful development and validation of our new LAMP assays, the next step was to use the assays on groundwater from contaminated sites. For this, we obtained groundwater from multiple contaminated sites across the US, as shown on this slide. Our work targeted the reductive dehalogenase genes, TCEA and VCRA, and the experimental design involved extracting DNA from triplicate groundwater samples and performing qPCR and LAMP on the DNA extracts. The results for TCEA are shown on this slide. The names of the groundwater monitoring wells are shown on the x-axis, and the y-axis illustrates the log gene concentration of TCEA. The average values obtained from qPCR are shown in the red bars, and those for LAMP are shown in the green bars. The black bars represent standard deviations from triplicate groundwater samples. Overall, you can see both methods produce similar results. The values were similar across multiple sites and over a range of gene concentrations. And we also find good replication between triplicate groundwater samples. This slide shows the results for VCRA. Again, the groundwater wells are on the x-axis and the gene concentration on the y-axis. This time, the qPCR results are shown in pink and the LAMP results are shown in blue. Overall, the results are comparable to those for TCEA. That is, similar results from both methods across multiple sites over a range of gene concentrations. Also, we find good replication among triplicates. To get a better idea of the comparison of the two methods over a concentration range, the data sets from the previous two slides are shown again here. The data for VCRA and TCEA have been combined. The values obtained using qPCR are on the x-axis on a log scale, and the values for LAMP are on the y-axis, again on a log scale. Overall, you can see that both assays produce similar values over a range of gene concentrations. So at this point, we were confident that our LAMP assays were producing comparable results to qPCR. So we started to think about possible applications for the assays. We were interested in exploring detection without DNA extraction because this would reduce the time and cost associated with the assays. We were also interested in using the assays with low-cost detection methods. We wanted to explore methods that could be potentially field deployable. This resulted in the development of what we have called a visual-based LAMP assay. The developed method involves only low-cost laboratory equipment, including a water bath and a benchtop centrifuge. The approach is based on the use of the fluorescent dye cybergreen, which binds non-specifically to double-stranded DNA. The dye is added following amplification, and the change in color is detected visually. This schematic is an overview of the visual-based LAMP assay. First, the groundwater sample is pre-filtered to remove material larger than 5 micrometers. Then the sample is concentrated onto a Cervex filter. An elution buffer is added. The filter is vortexed. Then the buffer and the cell mixture are removed. The mixture is centrifuged to concentrate the cells. Then the pellet is resuspended. Following this, the LAMP master mix is added. The vials are moved to a water bath for 1 hour at 63 degrees Celsius. Finally, cybergreen is added, and when the target is present, the solution changes to green, and that's shown on the left bottom of the slide. This approach was tested using both the VCRA and TCEA LAMP assays. Positive detections, as shown in green, were noted in different types of samples, including DNA extracts shown on the left, filter-eluted cells, as well as centrifuge cells. And then, as expected, the controls did not change color. We also examined the specificity of the LAMP assays. For this, we tested each assay using different plasmid standards. The image on the top illustrates the results for the VCRA assay, for the control, for the BVCA plasmid, for the TCEA plasmid, and for the VCRA plasmid. You can see only the VCRA plasmid produced a positive signal. A similar test was performed for the TCEA LAMP assay, shown in the middle of the slide, and the BVCA LAMP assay on the bottom of the slide. In all cases, a positive signal was generated by the plasmid with the clone target circled in blue. These results suggest that the LAMP assays are specific to their target genes. The next set of experiments evaluated the detection limits of the visual-based assays. This first involved using triplicates of a dilution series of the VCRA plasmid standard. We used a range of concentrations spanning from 10 to the 5 to 10 gene copies per reaction. The controls are shown on the left, and the 3 rows represent the triplicates of the dilution series. The difference in color was shown at 10 to the 2 copies per reaction, and similar results were produced for both TCEA and BVCA. At this point, we were confident that the assays were specific to their target sequences and were sensitive enough to be of practical use. Therefore, the next step was to test the assays using groundwater, and we did this without DNA extraction. We obtained groundwater from 4 sites and tested multiple samples from each site, each in triplicate. We also quantified the target gene using qPCR on DNA extracted from groundwater. And so the results are shown on this figure. On the x-axis are the groundwater wells, and on the y-axis are the values obtained using DNA extraction and qPCR. And so the green bars are those that showed a positive detection with the LAMP-based visual assay without DNA extraction, and the orange bars are those that did not change color. And so the threshold for positive detection with the LAMP assay appears to be approximately 10 to the 5 gene copies of the VCRA gene per liter. Therefore, if a site has 3 replicates that remain orange, then bioaugmentation or biostimulation may be necessary, as typically more than 10 to the 5 gene copies per liter are required for effective dechlorination. A final set of experiments combined the visual-based LAMP assay with the approach known as most probable number, given the acronym MPN. We combined the methods because we wanted to make the visual-based LAMP assay quantitative. And so the results are shown on this slide. On the x-axis are the results from most probable number LAMP, and on the y-axis for qPCR. We gathered data for both VCRA as well as TCEA. And overall, you can see a positive correlation between the 2 methods, indicating that LAMP most probable number would be a viable quantitative approach. Before I move on to the next topic, I wanted to take some time to distinguish between 2 sequencing approaches. 16S RNA amplicon sequencing is probably the most commonly used approach. It targets a region of the 16S RNA gene, therefore providing taxonomic information about microbial communities. It is used to determine which microorganisms are present. For example, is Staphylococcus McCartyi present? Another method, shotgun sequencing, is less commonly used on groundwater. This involves whole genome sequencing, where random DNA strands are sequenced. The method can provide information on which functional genes are present. For example, what is the relative abundance of BCRA or ETNC? In fact, it has the potential to provide data on a limitless number of target genes. It can also generate relative abundance information on which microorganisms are present. So in this project, we used shotgun sequencing to examine the microbial communities at 5 chlorinated solvent sites. For this, we targeted both the functional genes associated with contaminant biodegradation, as well as the relative abundance of non-chlorinated solvent degraders. The research involved the analysis of 20 groundwater samples, and as I said, from 5 chlorinated solvent sites. And each site had previously been bioaugmented with the mixed culture SDC9. The chlorinated solvents and or their degradation products were present at these sites, and at one site, 1,4-dioxane was present. The approach involved the sequencing platform Illumina HiState 4000, which generated between 4 and 5 million sequences per sample post-quality control. The sequencing data was then analyzed using 2 methods. We performed a taxonomic analysis using MGRAST, an online analysis server. This provided data on which microorganisms were present in each sample. However, classification is only recommended to the genus level. The second approach involved functional gene analysis. The first step here was to collect known sequences for the functional genes of interest. For this, we collected FASTA sequences from FunGene, and this is an online gene pipeline and repository developed here at Michigan State by the researchers at RDP. We also collected sequences from NCBI. Following this, the databases for each gene were then aligned against our shotgun sequencing data using the software known as Diamond. Moving on to the results of the taxonomic analysis, this slide illustrates the relative abundance of genera previously associated with chlorinated solvent degradation. The 6 graphs include the data for the 5 sites and also for SDC9, shown on the bottom right. Between 2 and 6 groundwater samples from each site were analyzed. The genera of known degraders are shown on the x-axis and the relative abundance on the y-axis. Dihalococoides was detected in every sample at every site, although in some cases the values were low. The abundance of dihalococoides was greater, not surprisingly, in the injection wells compared to the monitoring wells. We find disulfidobacterium at all 5 sites, although the relative abundance was typically less than that of dihalococoides. At 3 sites, Geobacter was the most abundant genus in this group, and at 2 sites, it was either the 2nd or the 3rd most abundant. Again, however, it is important to note that this analysis is only at the genus level. Therefore, it may overestimate the abundance of possible degrading microorganisms. Moving on to the functional gene analysis, this graph indicates the relative abundance of 4 genes encoding for reductive dehalogenases from Dihalococoides mcchordii in these groundwater samples. The names of the wells are again shown on the x-axis and relative abundance on the y-axis. You can see in the majority of cases, TCEA and VCRA were the most commonly detected reductive dehalogenases. We also detected reductive dehalogenases from Dihalogenemonas, Dihalobacter, and Disulfidobacterium. However, these genes were found with much lower relative abundance values. Although the MGRAS data indicated Geobacter and Polarimonus were present at all sites, and this is data from the previous slide, we find that reads aligning to PCEA of Geobacter lovelii and cytochrome P450 of Polarimonus JS666 were observed only from 1 sample each. These findings emphasize the importance of functional gene analysis to more clearly define the in situ potential biodegradation capabilities. The relative abundance of genes associated with aerobic chlorinated solvent degradation was also investigated. The graph on the left indicates the relative abundance of ETNE and ETNC, and the graph on the right indicates relative abundance values for PMOA and MMOX. Again, the names of the wells are on the x-axis. Overall, you can see the values for all 4 genes differ across the samples. In many cases, MMOX was more dominant compared to PMOA. Notably, these aerobic genes were found in at least 1 sample from each site. And so aerobic and anaerobic functional genes were detected together. However, it is important to note that the highest values for ETNC and ETNE were 2 orders of magnitude smaller than those for VCRA or TCEA. The data sets were also searched for the functional genes associated with 1,4-dioxane biodegradation. We focused on the 12 genes summarized in a recent publication, and that's shown on the bottom of the slide from ES&T Letters from 2017. These genes were linked to both metabolic as well as co-metabolic degradation of this chemical. This time the data are shown in a slightly different format. On the x-axis is the sum relative abundance which considers all of the groundwater samples collectively, and the functional genes are shown on the y-axis. From the 12 genes examined, only 6 were found in the groundwater samples. Interestingly, these genes were detected in at least one sample from all 5 sites, despite the fact that only one site was known to be contaminated with 1,4-dioxane. Another obvious trend is the dominance of the gene from methylocyanus trichosporium, OB3b, shown at the top of the slide with the blue arrow. Also, the genes from the 2 Pseudomonas strains and Burkholderia capacia, G4, were detected across a number of samples, also at the top of the slide. Surprisingly, the functional genes associated with the 3 Pseudonocardia strains or the 2 Mycobacterium strains were not detected. We also analyzed the MG-RAS data to determine the relative abundance of genera previously associated with 1,4-dioxane biodegradation. Again, it is important to note that the classification is only to the genus level. Therefore, we do not know if specific species or strains are present. The sum relative abundance is shown on the x-axis and the genera investigated on the y-axis. In this case, Pseudomonas and Burkholderia were the most dominant, shown by the blue arrows. It is interesting to see that Mycobacterium was present, even though the 1,4-dioxane degrading functional genes associated with this genus were not detected. This, again, perhaps emphasizes the importance of functional gene analysis over taxonomic analysis. On the other hand, agreeing with the functional gene data, reads aligning to the genus Pseudonocardia were not detected. Some key points then regarding the shotgun sequencing data. It was interesting to note that reads aligning to both aerobic as well as anaerobic biomarkers were detected across all sites, although the relative abundance values were higher for the reductive dehalogenases. In some cases, reads aligning to both reductive dehalogenases and 1,4-dioxane were present. This could suggest the ability of these communities to degrade both contaminants if the correct conditions were created. Finally, shotgun sequencing generated a greater depth of information compared to current methods, such as 16S RNA sequencing or quantitative PCR or LAMP. However, several issues still need to be resolved before this approach can be widely adopted. These include the need for a comparison of relative abundance values to values generated with qPCR. We generated preliminary data indicating a positive correlation between the 2 methods for VCRA, which I didn't have time to talk about today. However, additional data is needed for other functional genes and also for a wider range of gene concentrations. Also, the approach can be expensive. Our cost was approximately $210 per sample, and there may be some issues regarding the availability of the method. Overall conclusions for the project. The LAMP assays designed for the targeted genes produced similar results to quantitative PCR. Similar values were generated using DNA from the 2 bioaugmentation cultures, as well as DNA extracted from groundwater samples from multiple chlorinated solvent sites. We find that the LAMP assays were less sensitive to inhibition by humic acid compared to quantitative PCR. And this issue could be important for groundwater samples with higher levels of organic material. We developed a visual-based LAMP assay using the fluorescent dye cybergreen. The assay only requires low-cost laboratory equipment, including a water bath and a benchtop centrifuge. Further, the assay could be used without DNA extraction to determine if VCRA gene copies could be high enough for efficient dechlorination. The assay was also combined with the most probable number approach so that it could be used in a quantitative manner. And then the most important take-home message about the shotgun sequencing analysis being the potential to examine a limitless number of functional genes. The methods described in this webinar have a number of benefits to the DoD. The molecular tools developed should provide a greater understanding of the underlying mechanisms for contaminant biodegradation. This is of value because of the lower cost associated with natural attenuation, biostimulation, or bioaugmentation compared to other remediation methods. The tools developed in this research can be used to examine biodegradation trends across sites, across time. The molecular data generated can then be combined with contaminant removal trends to provide a more complete picture of site conditions. The LAMP assays offer a low-cost and potentially field-deployable approach for monitoring bioremediation at DoD sites. And then finally, shotgun sequencing will be a better tool to address site cleanup when multiple contaminants are present, as is the case at many DoD sites, because the approach can provide relative abundance values for numerous genes associated with the biodegradation of numerous contaminants. Finally, I would like to acknowledge the co-PIs on this project, Syed Hashim and Robert Stedfeld, as well as Paul Hatzinger from Aptim. Thanks also to the students that, the students that were part of this project, Hong Yu Dang and Yogendra Kanatar. Also thanks to Phil Dennis and Frank Loeffler. A big thanks to a list of people from Aptim for collecting and sending the groundwater samples. And then thanks to a list of researchers from MSU for helping with the analytical methods and also with the functional gene analysis. Additional information, including the papers produced by this work, can be found on the website listed on this slide. Thanks for your attention. I'd be happy to take any questions.
Thank you, Allison, for a very interesting presentation. We've received a number of questions that we would like you to answer. But as a reminder for our audience, please submit your questions by using the Q&A box on your screen. All right, the first question received is from Colorado State University. With 6 primers, how is LAMP affected by gene variants?
That's a good question. And so we have to be very careful when you design the LAMP primers. For the reductive dehalogenases, we collected all of the sequences in NCBI. We designed them very carefully. But for genes that have more variants, then it may not be as an effective method because you've got this high level of specificity. So I would say it's very dependent on your gene sequence, and I would just look into the databases and carefully design the LAMP primers for that.
Thank you. And a follow-on question, can you incorporate a reasonable amount of degeneracy in your LAMP primers?
I am not so familiar with those techniques. I would have to get back to you on that answer, but I don't want to answer it and be totally off base on the question.
Understood, and we'll provide you the name of the person that asked you this question.
Thank you.
All right, another question. On slide 26, which humic acid was used in your experiment?
For that, I would have to go back to the original paper and see where we purchased that humic acid from. And so we have a couple of papers that were published in the Journal of Microbial Methods, and where we bought the humic acid should be listed there.
Great, thank you.
Those papers should be listed on that the website, and I can also provide that information to the person asking that question a little later.
Thank you. Is LAMP applicable, or can it be adapted to transformation of other contaminants beyond the ones that you described in this presentation?
Yes, I think it could easily be used for other contaminants, for the functional genes associated with other contaminants. To date, it's primarily been used for pathogens, for organisms that are of threat to human health. And so I haven't seen any other examples in the literature for genes associated with the biodegradation of contaminants, but there's no reason at all why it couldn't easily be adapted to those genes. The primer design software is very easy to use.
Thank you. And a broader question, can you describe or talk about whether there are any major disadvantages to using LAMP compared to more traditional techniques like qPCR?
Yes, thanks for that question. I meant to mention this in the seminar. So a major disadvantage with LAMP is the potential for the volatilization of the amplicons that are generated. And so this is a more important issue for LAMP compared to qPCR because of the larger number of amplicons. And so, of course, this is problematic because it has the potential to cause cross-contamination between samples. So this would, of course, create artificially high numbers and the potential for false positives. However, we have found that you can avoid this issue by making sure that you do not open any vials or any plates following LAMP amplification. This ensures that you don't get any volatilization of those LAMP amplicons.
Thank you, Allison. With regards to the shotgun sequencing data, how easy is it to analyze it, and what improvements could you make to your shotgun sequencing approach?
And so the MG-RAST analysis for the taxonomic profiles is very straightforward. The data files are simply uploaded onto the MG-RAST server, and then it may take a few weeks for the automated analysis to be complete. And so the pipeline automatically merges paired-end reads, trims low-quality regions, and removes artificial duplicate reads. And then the RefSeq database is used to generate taxonomic profiles. And then the classification files are simply exported into Excel for additional analysis. Although, again, the major disadvantage here being that the classification is only to the genus level. And so for the functional gene analysis, probably the most time-consuming step is the collection of the FASTA files for the genes of interest. Although once these databases have been generated, have been collected, you can use these for all future studies. And then the FASTA files generated are examined for low-quality sequences using Trimmomatic, and the software also removes adapter sequences. Following this, the analysis is performed with Diamond, and that's relatively easy to use. It only requires a few command lines. And then the data sets from Diamond can be manipulated within Excel. And so you can sort for alignment, identity, and length, and counting the number of reads that fit those criteria. And finally, the relative abundance can be determined. And so, sorry, did you have another part of a question that you mentioned?
No, let me just ask you one quick question about technology transition before we move on to Mandy's presentation, and then we'll get back to any remaining questions during the final Q&A. What are the remaining steps that are needed before LAMP can be deployed in the field?
Probably the most limiting step is a detection device, an automated detection device. And once that's developed, then it should be relatively easy to perform LAMP within the field. We developed this visual-based LAMP assay, but there are other ways to detect LAMP amplicons that are not visual but can be automated, and that is probably the biggest step going forward for the use of LAMP in the field.
Thank you, Allison. And again, we'll get back to any remaining questions for you in the final Q&A. And at this point, we're going to transition to the second portion of today's webinar, which will be delivered by Dr. Mandy Michelson. Mandy is a research engineer with the U.S. Army Engineer Research Development Center in Seattle, Washington. Her current research areas focus on novel applications of groundwater remediation technologies to accelerate cleanup of explosives and chlorinated solvent contaminated aquifers. Since joining the U.S. Army Corps of Engineers in 2008, Mandy has served as a PI and lead engineer on field-scale technology demonstrations and full-scale groundwater remedy optimization projects. Mandy received her bachelor's degree in civil engineering from the University of Iowa, and both her master's and doctoral degrees in civil engineering from Oregon State University. Dr. Kate Kusharzek is the co-PI for the research that Mandy will describe today. Kate is a principal research scientist with Battelle in Columbus, Ohio. She has served as a PI on projects focusing on applying and monitoring bioremediation processes at contaminated sites. Her project experience and fields of expertise include soil, groundwater and sediment remediation, biochemical basis of bioremediation of munition constituents, and Humic technologies and performance monitoring. Kate received her master's degree in biochemistry from Marie Curie University in Poland and a doctoral degree in environmental microbiology from the University of Idaho. And Kate will be standing by to answer questions following Mandy's presentation. Mandy, please proceed.
Thank you, Rula, and thanks, Rula and Andrea, for the opportunity to give this webinar today. Tens of thousands of sites within the DoD complex are contaminated with chlorinated solvents or chlorinated ethanes. And special bacteria called D. halicoides have the capability to respire these chlorinated ethanes to form non-toxic ethane. And so the abundance and activity of these special bacteria is quite important for management of these chlorinated solvent contaminated groundwater sites. Quantitative PCR is an existing molecular biological tool that tells us about the abundance of these bacteria at the sites. But the motivation for this project was that while the gene abundance for these bacteria tells us about the activity potential for reductive dechlorination, we hypothesized that the abundance of the reductive dechlorination proteins, or the RDA's proteins, which is that genetic activity expressed, would provide us with potentially a better proxy of reductive dechlorination activity in situ. So the objectives of this laboratory demonstration were to quantify the reductive dechlorination rates in a series of microcosms, and also to quantify the RDA's abundances in those microcosms, to quantify the correlation between the RDA's biomarker abundances and the reductive dechlorination rates, to develop a quantitative, a predictive model that would allow one to predict reductive dechlorination rates based on biomarker abundances, and then finally to empower end users to use the quantitative proteomics tool, which is the way we quantify these proteins as an additional line of evidence in support of M&A assessments, and also management of bioremediation at these sites. On slide 54, this idea is really exemplified. So this is an outcome of the BioPIC project, which was an ESTCP project of the year in 2015, I believe. And so that project went out to sites, chlorinated solvent sites all across the nation, quantified the reductive dechlorination rates and the D. halicoides or DHC cell abundances. And we see a plot of those two. The DHC abundance is on the x-axis and the rate coefficient for cis-DC is on the y-axis. There's a lot of spread in this data. So really our motivation was if we had plotted the rate coefficients versus protein abundances, we hypothesized that relationship would be tighter. So on slide 55, our first order of business was to select SDC9 as the reductive dechlorination consortia we'd focus on during this laboratory demonstration, because a lot of sites across the country have used SDC9 consortia for bioaugmentation, and so our results would be relevant right out of the gate, and also to keep it simple. And so we sequenced the metagenome for SDC9 and identified 14 unique RDA's genes that otherwise wouldn't be predicted just by querying the databases. And so it was good that we sequenced the metagenome. And this is also an important point to clarify nomenclature. So a gene sequence is related to a genome, and it's a genetic sequence, whereas a protein, a peptide is analogous. So a peptide is a sequence of the entire protein. And for this project, when we say protein abundance or proteins per mil or peptides per mil, there's a one-to-one correlation in that there's one unique peptide sequence per protein of interest. So on slide 56, there's a schematic of the proteomics approach that's identified. So after identifying the unique RDAs genes that we wanted to focus on, we could annotate the corresponding proteins and identify the unique peptide sequences for each of the proteins. And so 39 candidates were identified. And Kate, who's on the line, and her team at Battelle ordered up isotopically labeled versions of those peptides that could be used as internal standards in the qProt assay for quantifying the absolute abundance of those peptides present in a given sample. And so on slide 57, a total of 4 proteins were identified to carry forward. PCA, TCEA, and BCRA are all reductive dechlorination RDAs proteins, whereas FDHA is a formate dihydrogenase proteins that's not specific to chlorinated ethene respiration, but is carried by the DHC cells. It's associated with how these cells make a living, energetically speaking, and so that protein was included as well. And so the number of unique peptides for each protein is shown in the ID column. The sequence associated with the peptide is shown, and the triplicate method detection limit results for these peptides is shown as well. And so ultimately the highest of the MDA results for each of the peptides was selected as the method detection limit for this study. On slide 58, at this point, we had the detection limits quantified through the detection limit study, and so now we sought to validate those results. And we did so by using a SDC9 stock solution, which we sequentially diluted to achieve 10 to the 7, 6, and 5 DHC cell titers. And then we subjected samples from those to the qProt assay. On slide 59, we see the results of that validation study for all of our 4 proteins. The green highlighted peptides are the quantifier peptides for each of the proteins, and those had the best sensitivity for quantification. The others are considered qualifiers, which are analyzed and quantified, but aren't used to estimate the protein abundance in the sample. And so where multiple quantifiers exist for a given protein, the results or abundance of the 2 peptides present in that sample are averaged to estimate the protein abundance in a given sample. And so on slide 60, we had good results, good quantitative results for the 10 to the 6 and 10 to the 7 DHC cell abundance samples, which was positive. As expected for the 10 to the 5 DHC cell abundance titer, we had mostly non-detect results. And that's because for the qProt assay, a total of 10 to the 6 cells in the sample, total cells needs to be present and extracted to be above the detection limit. And so in this case, at 10 to the 5, given that we had a 10 to the 5 cells per mil, and given that we had a 15 mil sample size, we were just over that 10 to the 6 total cells present in the sample. So getting mostly non-detect results for that sample wasn't completely unexpected. So then on slide 61, we expressed the results that we achieved in both protein concentration, femtomole protein per milliliter units, and also in protein per DHC cell concentration units, and those are highlighted in red. For FDHA, TCEA, and BCRA, which are all carried by the dihalicocoides cell that we're interested in. PCEA is carried by other organisms that are present in the SDC9 consortia, but not by DHC. So that relative concentration, protein per cell, is not provided for that protein. But we wanted to do this extra step to ensure that we didn't have any suspiciously muscular, so to speak, DHC cells present, and to ensure that the protein per cell results we were achieving were within range of other published studies, and they were, which was positive. So now with the quantitation piece completed, we moved on to the microcosm study. So on slide 62, we collected materials, aquifer solids, and groundwater from Joint Base Lewis-McChord, which is a landfill located near Tacoma, Washington. It's a chlorinated solvent contaminated site. And on slide 63, the microcosm study schematic is illustrated. We had live and killed controls. Some microcosms featured groundwater only. Some featured a mixture of groundwater and solids together. We added 500 milligrams per liter lactate as the growth substrate. We spiked with a range of SDC9 consortia to achieve a range of DHC cells per mil in the microcosms. Calcium carbonate was included as a pH buffer, and cis-DCE was spiked in at approximately 10 milligrams per liter. And then samples were collected over time to both quantify the reductive dechlorination rates and also to quantify the biomarker abundances, both genes and proteins. We initially considered including transcripts as in addition to gene abundances in this study, but had difficulties with that analysis, and so that was ultimately excluded from the study. On slide 64, we have an example of what the results looked like. It gives you a sense of the change in the CVOC or chlorinated ethane concentrations over time during the study. The red is cis-DCE, and then the vinyl chloride is shown as green and yellow as ethane. And so with the range of cell abundances, DHC cell abundances, we achieved a range of activity and a range of cis-DCE and vinyl chloride first order rate constants, which are shown in the table below. On slide 65, a snapshot of all of the rate coefficients that we obtained are shown. We obtained a total of, there were 44 total microcosms that were completed, and so here we're just showing a subset of the different microcosm sets and the rate coefficients that corresponded to those sets. We screened to determine if the rate coefficient was acceptable for a given microcosm by looking both at the global R squared of the model fit, as well as a combination of the 95% confidence interval on the K-cis and KVC rate coefficient values. And together, if both were acceptable, then the microcosm was considered acceptable for carrying forward into correlation analysis. So on slide 66, the results of the correlation analysis are shown. And we did that two ways. In the upper part of the table, the correlation analysis between the rate coefficients and the protein and gene biomarkers for all of the time points are shown because we collected an early, initially following bioaugmentation, a midpoint, and at the conclusion of the microcosm study. And so we looked at the correlation with all of the biomarker time points, again, that's up top, and also just with the midpoint biomarker abundance only, and that's in the bottom part of the table. Slide 67 illustrates that looking at only the midpoints showed a significant improvement in the correlation results for all microcosms for both the K-cis and the KVC values. So for example, if we look at the correlation between log K-cis and vinyl VCRA protein, we increased from 0.374 to 0.725. And so because all of the correlations improved, which made sense to us in terms of the midpoint being an integrated biomarker abundance and the rate coefficient being an integrated measure of the activity, we elected to move forward with the regression using only the midpoint biomarkers for the microcosms. So slide 68 shows the regression results. The upper plot is for K cis-DCE, the bottom is for KVC versus the biomarker abundances. Gene abundances are shown to the left in green, purple, yellow, and gray marker colors. And for proteins, they're shown to the right on the plots in red, blue, and orange colors. And so the power law least squares regressions results were good. All regression equations featured a R² of 0.78 or greater. And the vertical red line that you see corresponds to the detection limit for proteins in protein abundance per milliliter units. And so that's at 1 × 10⁶ proteins per milliliter. So extrapolating the regression results down closer to that detection limit for the samples and then going over to the y-axis translates to a rate coefficient associated with the detection limit of very low, 0.04 or so inverse years, which is certainly low enough to be relevant for monitored natural attenuation assessments at chlorinated solvent sites. So that was positive. On slide 69, we now looked at the data overall to say, okay, how good is our prediction capability? How well can we use biomarker abundances to predict reductive dechlorination rates? And we did this in 2 ways, using data that was not featured to produce the regression equation, but for microcosms that were passed our quality checks for both the rate coefficients and also the biomarker abundances. We did it in 2 ways. We randomly picked microcosm biomarker abundances that corresponded to 10⁶, 7, or 8, and then used those biomarker abundances, input them to the regression equations, and predicted rate coefficients. Or we looked at the global average of the biomarker abundances over the range of DHC cell concentrations, then use those average biomarkers, input those abundances into the regression equation, and predicted the rate coefficient. And so the results for KVC, or vinyl chloride, are shown here. And so the black bar is the actual measured rate coefficient for that given data set, either randomly picked or the global average. And the green bar that surrounds all of the bars within a given set reflects the uncertainty around that measured rate coefficient. And the take home here is that regardless of how you test the data, either randomly picked or global, the gene abundances and the protein abundances do a pretty good job. When you look at the random microcosm set, the protein abundances tend to overpredict the rate coefficient for vinyl chloride, especially at the higher DHC cell abundances. And when you look at all of the biomarker abundances together on average, and that's the green bar, the condition tends to improve. And when you look at the global average overall for all of the microcosm data sets, the proteins and the genes in terms of their predictive capability for rate coefficients are on par with one another. So on slide 70, a couple of things to consider here is that although both genes and proteins did a decent job in this study, which was a positive finding in terms of their rate predictive capability, we know based on published literature that elevated concentrations of DHC gene abundance doesn't necessarily translate to good ethene production in the field. And likewise, elevated abundances of functional genes, of reductive dechlorination functional genes in proportion to DHC cell abundance likewise doesn't necessarily translate to good ethene production in the field. And so what we hope to do now and continues to be our hypothesis is that at field sites, under realistic field conditions, thinking back to the biopic results, that protein abundances will be just as good or even better at predicting the reductive dechlorination rates in the field. And so we hope to embark on that next piece of the project in the field very soon. On slide 71, a key question for this project and this technology is, can you quantify proteins low enough to be relevant for M&A assessment, which is a really important piece of chlorinated site management at DoD sites. The first two rows of the table shown here for 10 to the 7 and 10 to the 5 cells per mil treatments translated to a rate constant of and this is for cis-DCE, rate constant of 580 or 8.4 per year. But a key consideration is that because this was a laboratory demonstration, we were limited on the total amount of sample volume that we could collect. And so in this case, it was a 15 milliliter sample volume. In the field, of course, we'll be able to collect orders of magnitude more. For example, a typical groundwater sample volume is on the order of a liter. So 2 orders of magnitude more sample to extract, digest, and analyze. And so that because we can collect more cells in this larger volume, it would allow us to digest more cells and see even lower in terms of the cell abundance. So these cell concentrations that we analyzed during our during the laboratory study would translate in a field setting to orders of magnitude lower in terms of rate coefficients. So on slide 72, what that looks like in terms of the biopic results are shown. You can see the 8.8 and 0.1 inverse year rate. That level of activity is certainly low and within range for M&A assessments. And we were, even in those cases, that 0.13 rate coefficient corresponds to a sample that was still orders of magnitude above the detection limit for the, in this case, TCA protein abundance at the site. And so the take-home message shown on slide 73 is that the results of this study really showed that we can quantify the proteins associated with reductive dechlorination, and that we can quantify them at low enough levels to be relevant for M&A site assessments, which was an exciting finding of the study. So on slide 74, the key findings are, number 1, that we confirmed that we can quantify the RDA genes and proteins at environmentally relevant M&A range abundances of DHC. We confirmed the positive and significant correlation between the RDA genes and protein biomarkers, and further quantify that relationship via regression analysis, and demonstrated the predictive power of gene and protein biomarkers in this laboratory study. And so the next steps involve taking this to the field. And we're really excited about this, the next phase field demonstration, because multiple field sites will participate in the study, some of which will have an ongoing M&A assessment occurring in parallel. So we'll be able to document the methods used to estimate the rate coefficients at the sites, the relationship between the biomarker abundances and the field scale rate constants, and also the interaction of the regulators with this technology along the way. On slide 75, the project team, outstanding team, of course, Kate Kushavik and Jada Meisel at Battelle. Kate's around to answer questions at the end. Paul Hatzinger with APTIM, Frank Loeffler and Fadime Kara Murdoch at University of Tennessee, John Wilson at Scissortail, and Jack Istock at Oregon State University. Thanks on slide 76. Many thanks to Carlotta Salucci, who provided materials from a Navy site. Also Kathy Gerber, who provided materials from a Vandenberg Air Force Base site. And to Meseret Galabrisi from JBLM, who provided materials that were featured in the Microcosm study. Happy to take any questions. Thank you.
Thank you so much, Mandy. We have a number of questions for you. Can you let us know when your described technology will be commercially available?
Yeah, Kate Kushavik is on the line and can talk more specifically about availability of proteomics.
Sure. Thanks, Mandy. So the Patel and Microbial Insights have been working together to bring this technology to market, especially for our commercial clients as well as the governmental ones. So this technology is already available for the end user. And if you would like to submit samples to us, we encourage you to connect through Microbial Insights so that we can take any orders. So the targeted quantitative proteomics that Mandy talked about, that's available, as well as shotgun discovery proteomic analyses that we can do for other contaminants of interest to understand global composition of proteins in the proteome in our microbial samples. Yep.
Great, thank you, Kate. Mandy, a second question for you. How likely are proteomics results going to be accepted by regulators as an additional line of evidence for M&A at this time?
Yeah, Kate Kushavik and I have had some good discussions about that, and she has a couple of project-specific examples she can provide where proteomics were part of the regulatory decision-making.
Right. So we had conducted 2 studies in which regulator was fairly happy to see the data from. One of them was at the gasoline contaminated site where we had done comparative assessment of degradation biomarkers using proteomics as well as metagenomics. And some of this data provided additional line of evidence to an occurring M&A of a contaminant. And the second site where proteomics was applied to confirm M&A of explosives also helped to navigate the applied technology that helped to drive concentration of a contaminant down and provided, again, this additional line of evidence that not only microbes that convert a contaminant were present, but they were also active and actively utilizing that specific contaminant of interest through the proteomic analysis. So we believe that this is a really valuable line of evidence in terms of M&A, but also sites that are undergoing active remediation. There are no firm guidances in terms of proteomics and in terms of interpretation of the data, but guidance exists in terms of application of mass spectrometry at sites and application of protein extraction protocols. So we're still at the phase of waiting for specific documents to be developed for the environmental practitioner in terms of application of proteomics. That could be readily applicable to sites, and that could be cleared by the regulator.
Great, thank you both. How applicable is your approach to potentially other contaminants that may be present at DoD sites? Mandy?
Yeah, I think, well, Kate, I think is probably the best person to field this one also.
So what was the question again, Roland?
How likely is your approach possible or applicable to other contaminants that may be present at DoD sites?
Right, so at Bateau, we are also following proteomic analyses for PCBs, for explosives such as RDX or TNT. We have been looking at common herbicides, pesticides, etc. So there's a variety of different contaminants that if a microbial community exists and can degrade, we will be able to develop an approach for that specific contaminant. One for dioxane is also a good example here. So in terms of global proteomic assessment, it can be done for nearly anything that is biodegradable. For quantitative targeted proteomics, this approach can be also developed as a method for any contaminants that are degraded by microbes, and we already know a suite of specific enzymes that can break down that contaminant of interest.
Great, thank you. Will there be a tool that end users can utilize to predict rates based on protein abundance in a sample?
Yeah, thanks, Rula. So the follow-on study that we have planned, which I described briefly, again involves going out to, we have 3 candidate sites identified, field sites, one of which is undergoing a monitored natural attenuation assessment that will occur in parallel, which is really great, a great site to include in the study, where multiple biomarkers, genes will, and also some stable isotope work is going to be done as well as rigorous plume assessments. So we'll work along on a parallel track with that site using the historical site data to estimate field scale rate coefficients. And then also at 2 other sites as well, we'll gather the data and perform the rate coefficients per standard EPA guidance, and we'll publish the methods for evaluating the rate coefficients at the site and the overall plume stability. And then we will collect groundwater samples all throughout those 3 sites, multiple samples, and we'll submit those to Kate for Kate's analysis, qProt analysis. So we'll have those RDAs, protein abundances, and then we'll relate all of that data together, and we'll perform a regression analysis that will allow folks to compare a biomarker abundance collected from their site in context with the activity versus biomarker abundance that we established through this 3-site study. So that's one way that folks will be able to use I suppose you could call that a tool, an instrument that folks can use to interpret their biomarker abundance data with respect to rate coefficients. Those folks can also use the detailed report that comes out of that second phase as a guidance document for estimating rate coefficients and assessing plume stability at their site. So yes, I think is the answer. We haven't developed that yet because we're just embarking on the field component of the demonstration, but it will be available in about 2 years.
Great, thank you, Mandy. And at this point, I'd love to pull Allison back in the discussion before we wrap up here. So Allison, we'll start with you. What final message would you like to leave the audience with today regarding the use of advanced molecular biological tools in groundwater contaminated with chlorinated solvents? Can you please get us started?
Sure. I think probably one of the main considerations going forward is the issue of the value of quantitative approaches when it comes to shotgun sequencing. So I think there's still a lot more work to be done in providing evidence that it can indeed provide quantitative information. And so, going forward, that would be comparing results from quantitative PCR for each of these functional genes compared to the data that we get from shotgun sequencing over a range of different site conditions, soils and sediments and groundwater. So that's one issue, making sure that quantification is robust enough to use. And then another major issue when it comes to shotgun sequencing will be the detection limits. We all know that quantitative PCR, their detection limits is impressive. What kind of detection limits will we get when we compare quantitative PCR to shotgun sequencing? And so going forward, that's a few areas that we're going to look into.
Great, thank you, Allison. Mandy and Kate?
Yeah, I'll start here. I think something that was really exciting, that was an outcome right out of the gate from this laboratory demonstration is showing in a very quantitative, robust way that we can quantify the proteins that are associated with or responsible for reductive dechlorination at chlorinated solvent contaminated sites. That hadn't been previously done before. Kate and her group, and part of the folks that were on this project team, have a publication that described the methods used to quantify these proteins, and that's under revision and will be in press very, very shortly, that came out of this project. And so we'll update the website to include that citation. We also showed that we can quantify those proteins at a low enough abundance to be relevant to M&A assessments. And so that's exciting, because that means, call up Kate and her colleagues at Microbial Insights. You can submit a sample from your site, quantify these proteins, and provide perhaps not a direct rate assessment at this time, because that's still somewhat under construction, given our field phase hasn't launched yet. But that to be able to quantify those proteins at low abundances right now, that's available right now, can provide an additional line of evidence for M&A assessment right now. And to me, that's an important take home.
Great, thank you. Kate, would you like to add anything?
I think I agree with what Mandy said. Another thing that is very exciting for us in this project is looking at potential additional targets in terms of protein biomarkers, as we are embarking on this M&A part of an adventure with proteomics, and looking at sites that have been either bio-augmented without a consortia or not bio-augmented at all, and understanding whether we can increase the decay of proteins that we've been using, or protein biomarkers that we've been using for our quantitative assessment. So that's definitely, from the perspective of more technically, analytically involving work, something we're looking forward to do.
Great. Well, at this point, we're going to go ahead and wrap up. Thank you to all of our phenomenal speakers today. And our next webinar in this series is on Thursday, September 19. It is in the Weapons, Systems, and Platforms program area, and will focus on tools for sustainment and health. This webinar will feature two speakers, Mr. Brian Hubbard from the U.S. Army's Joint Program Executive Office Armaments and Ammunition, and Dr. Andrew Henderson from Nobles. Registration is open, so please visit the CERDIP and ESCCP webinar webpage to register for this and other future webinars. Before we conclude, I would like to remind you that both the audio and a copy of the presentation of today's session will be archived on our webinar webpage in case you would like to refer to them in the future. We would appreciate it at this time if you could please take a moment to complete the survey that will pop up on your screen. This concludes today's webcast. Thank you so much for your time.